Next, we're going to talk about two important stereoisomeric relationships that exist between molecules. These are called enantiomers and diastereomers. The term stereoisomers itself describes a relationship between two or more molecules. In this section, we have been focusing on what are called configurational isomers. These are stereoisomers that differ in the RS configuration of one or more corresponding chirality centers. In general, if a molecule has n number of chirality centers, then it is possible that it can exist as a maximum of 2 to the n configurational stereoisomers. Now, some molecules will have fewer than the maximum number of stereoisomers because of mesocompounds, which will be stereoisomers that are not chiral. However, in general, if there are no mirror planes that exist in the molecule, there will be 2 to the n total stereoisomers of that molecule. Now, there are two types of stereoisomers when we have molecules that have chirality centers. The first is called enantiomers. Enantiomers are molecules. They have the same formula, they have the same connectivity. However, they are mirror images and they are not superposable. So, non-superposable mirror images. When we look at a given chiral molecule, there will only be one enantiomer of that chiral molecule because a given object can only have one mirror image. So enantiomers exist in pairs of two that are mirror images. In the same way that we have a left hand and a right hand, and we don't have some third type of hand. The other type of configurational stereoisomer that we see is called the diastereomer. Diastereomers are molecules that have the same formula and same connectivity. They're not superposable, however, they are also not mirror images. So they're non-superposable, not mirror images. A molecule can have many diastereomers. We're also going to see that molecules that have cis-trans stereoisomerism are diastereomers. So let's look at an example of these configurational isomers. We're going to start with this molecule right here. We're going to use the Fisher projection to compare these because it's much easier to see the relative configurations of the chirality centers and when they change. So looking at this molecule, we have a methyl group, 2 chirality centers, and then an ethyl group. So we have a total of 5 carbons in our parent chain. And then at carbon 2, we have a chlorine, and at carbon 3, we have a chlorine and a hydrogen. As drawn in the Fisher projection, the chlorine at carbon 2 is on the left, and the chlorine at carbon 3 is on the left, and then the hydrogens are on the right. We can make a mirror image of this molecule by imagining a mirror right here, and then imagining reflecting across that mirror. If you recall how a mirror works, atoms that are close to the mirror on one side should be close to the mirror on the other side. The effect of this then is that the hydrogen that's on the right here would reflect over, and it would be on the left in the mirror image so that it would stay close to the mirror. And the chlorine, which is far away from the mirror on the left side, would reflect over, and it would be on the right hand side. So it would be far away from the mirror. In fact, in the mirror image, every single pair of groups on each chirality center would be exchanged left and right. So chlorine and hydrogen are exchanged right here. Chlorine and hydrogen are exchanged right here. If we had other groups, we had more chirality centers, they would also be exchanged. To have a mirror image, every chirality center has to have its groups exchanged with each other. If we then look at this mirror image and we try to superpose it, recalling that we can only rotate it 180 degrees or slide it on the paper, we would see that this would become non-superposable. Remember, when trying to superpose Fisher projections, we are not allowed to pick them up, turn them over 180 degrees, and put them down. That actually creates the mirror image of the original projection. So these are mirror images that are not superposable. Because they are perfect mirror images, we say that they are enantiomers. Now let's look at what happens when we exchange the groups on the top atom. However, we leave the groups on the lower chirality center the same. In that case, this molecule is no longer a perfect mirror image of this molecule that we started with. However, once again, if we try to superpose these by sliding them on top of each other, we see that they don't perfectly superpose. The groups on this upper stereo center would not superpose upon each other. So what we have here is a pair of molecules that are not mirror images, but they're also not superposable. So we would call these two diastereomers. Similarly, we could exchange the groups on only the bottom chirality center. That would look like this, and the top chirality center would stay the same. And once again, these would not be mirror images, however, they would also not be superposable. So these would be another pair of diastereomers. So overall, what we have is, we have our original molecule, it has one enantiomer, and then it has two more diastereomers. We have a total then of 4 stereoisomeric molecules with this basic Lewis structure configuration. Because there are 2 chirality centers, we have 2 squared stereoisomers. 2 squared is 4. So these are the 4 stereoisomers of this particular Lewis structure, and there are no other stereoisomers of these molecules. The other thing to note is that if we look at, we started with this molecule in the upper left corner, we exchanged one chirality center, so we made a diastereomer. But if we look at that diastereomer, we can make a mirror image of that diastereomer by exchanging the top chirality center again and exchanging the bottom chirality center. And now these 2 molecules are mirror images of each other that are not superposable. So this is another pair of enantiomers. Both of these are diastereomers to our original molecule. In general then, what makes Fisher projections so useful is that we can really quickly tell if we have enantiomers and diastereomers. Because to make mirror images of molecules in Fisher projections, what we have to do is we have to exchange at the groups on the cross of every chirality center. If even one group is not exchanged, then that molecule will be a diastereomer of the original molecule. Some of this is summarized on the next page right here. All of the chirality centers of enantiomers have opposite configurations. In other words, the groups have been exchanged on the chirality centers. In contrast, only some of the chirality centers of diastereomers have opposite configurations. If any of the corresponding chirality centers in 2 different molecules have the same configuration, then they cannot be enantiomers. A molecule will have only one enantiomer. They exist as pairs. A molecule can have many diastereomers. Stereoisomers that have different configurations at chirality centers are called configurational isomers. It is possible to have enantiomers and diastereomers without chirality centers. We especially see this with diastereomers, where cis and trans molecules, like cis and trans double bonds, are examples of molecules that have the same connectivity, they're not mirror images, and they're not superposable. So those are diastereomers. One of the types of questions that you're going to be asked to answer is comparing two given molecules and determining the relationship between those molecules. We've already done this for connectivity. We're going to expand that to also look for stereoisomerism. This is a small flowchart that shows how we could work our way through that problem. We would start by taking two given molecules. In a flowchart, what we do is we ask a question and then we look at the possible answers and we sort of follow along the arrows as we go from possible answer to possible answer. The first question we want to ask when given two molecules then is, do they have the same molecular formula? If the answer is no, then the molecules are not any type of isomer because isomers are required to have the same molecular formula. We would just say that they are not isomers, or we would say that they are completely different structures or something like that. If they do have the same molecular formula, we would then ask, do they have the same connectivity? Again, if the answer is no, so they have the same molecular formula, but they have a different connectivity, we would call those constitutional isomers. On the other hand, if they still have the same connectivity, then we would ask, well, are they superposable? If they are superposable, then they're actually identical structures. They're not a type of isomer. They're exactly the same. On the other hand, if they're not superposable, then we just ask, are they mirror images? If they're not superposable, and they are mirror images, they're enantiomers. If they're not superposable, and they are not mirror images, then they are diastereomers. I encourage you to have this chart with you as you begin to work through some of the homework problems where these questions are asked. Work through the chart, and I think you'll be able to see that it becomes relatively straightforward to work out what the correct answer is. Once you've used the chart a lot, you'll probably eventually be able to do it really quickly without the chart. Remember that on our exams in the online class, we're going to have access to our notes. So you should definitely practice with the chart so that you can also use it quickly and efficiently on an exam.